Good morning to all. Hello to everyone joining today's webinar. I am Dr. Pradeep Bolani, and as the co-convener of this event, I am delighted to welcome a diverse and engaged audience. Your participation greatly enriches our discussions and underscores our shared commitment to advancing in our respective fields. I look forward to a productive conversations and collective learning as we embark on this educational journey together. SPSR is distinguished for its engaging webinars, further enhancing its status as a leading platform for continuous professional development in the pharmaceutical sector. I am confident that today's webinar will uphold our tradition of excellence, providing valuable insights from a respected speaker. Let us seize this chance to gain new knowledge, participate actively, and progress our field. Welcome, everyone. I look forward to a productive experience for all the participants. Thank you for joining us today. We are gathered today to commemorate the 98th SPSR webinar on data-driven drug design, merging pharmacokinetics, DOE and IP strategy for smarter R&D, a distinguished event hosted by the Society of Pharmaceutical Sciences and Research. Under the esteemed guidance of Dr. Shashi Alok, National President of SPSR, the Society continues to play a pivotal role in fostering innovation and advancement in the field of pharmaceutical sciences. The SPSR is devoted to advancing the pharmaceutical field through a variety of strategies. Our mission is to organize conferences that support pharmacy-related initiatives and endorse programs tackling public health issues in India, focusing particularly on disease prevention. We make it a priority to recognize and honor pharmacists whose notable contributions drive progress in our field. At the heart of our mission is commitment to spreading pharmaceutical knowledge, ensuring a steady and robust flow of information. By striving towards these objectives, we may aim to elevate the quality and impact of pharmacy profession, fostering a proactive and collaborative community dedicated to excellence. It is our great privilege to introduce Dr. B. Prakash Rao, Director R&D and IQAC, Moonray Institute of Pharmaceutical Sciences, Shadnagar, Telangana, India, as a distinguished speaker. Dr. B. Prakash Rao expertise greatly contributes to our event, providing substantial knowledge and insights. His participation is certain to enrich our discussion and expand exploration of data-driven drug design. We are grateful for his engagement and anticipate an enlightening session. It is our privilege to introduce Mrs. Monika Sabharwal as the convener for today's webinar, who also serves as the founder and National Secretary of SPSR and CEO SPSR Periodicals Private Limited. Her significant involvement and leadership in the pharmaceutical sciences and research communities are indicative of her commitment to the field. We are very grateful to have her guidance in today's session. Now we will start with the short introduction to guest profile. Dr. B. Prakash Rao is a highly accomplished academician, researcher, and pharmaceutical technologist with over 38 years of teaching experience, including more than 26 years of active research in the field of pharmaceutics. Currently, he is serving as the Director of Research and Development and IQAC at Moonray Institute of Pharmaceutical Sciences, Shadnagar, Telangana, India, where he plays a pivotal role in institutional planning, quality assurance, and guiding research innovation. In this leadership position, he is dedicated to promoting the academic excellence and strengthening the research culture through structured R&D initiatives and quality enhancement practices. His mentorship and guidance have been instrumental in shaping the careers of aspiring pharmaceutical professionals and researchers. Dr. Rao holds a PhD in pharmaceutics from Rajiv Gandhi University of Health Sciences, Bangalore. His academic foundation includes a b.pharm and m.pharm drug formulation and technology from Kakatiya University, Warangal. Throughout his academic journey, he has held several key positions such as the head of the department and principal in prestigious institutions, contributing extensively to academic and institutional development. He has supervised over 55 m.pharm dissertations and guided 11 PhD scholars with a strong focus on the drug discovery systems such as buccal and sublingual formulations, osmotic pumps, nanoparticles, SMEDDS, microparticles, floating DDS, and in situ gels. He is also proficient in advanced analytical and formulation tools and has expertise in software like Python, Design Expert, Kinetica, and JMP. Dr. Rao has published 69 research papers in high impact national and international journals, authored software for pharmacokinetic and BA predictions, and is credited with multiple Indian patents, including innovation in oral liquids for diabetes and cancer and in situ gel formulations. His work has attracted extramural research funding from government and private agencies promoting impactful translation research. A recipient of numerous awards, Dr. Rao has been honored with the Diamond Achievers Award, Asian Admirable Achiever Recognition, and Best Indian Golden Personalities Award. He has also proudly represented India at prestigious international platforms in Chicago and China. A visionary leader and a passionate educator, Dr. Prakash Rao continues to inspire the excellence in pharmaceutical education and research, contributing meaningfully to national and global pharmaceutical innovation. It is with great satisfaction and pride that we declare that we have exceeded 1,300 registrations for this webinar. Registrations are received from India, Algeria, Azerbaijan, Egypt, Ghana, Indonesia, Kenya, Kuwait, Malaysia, Nigeria, Oman, Romania, Turkey, Uganda, USA, and Vietnam. We are deeply moved by the substantial and varied responses we have garnered. Our heartfelt gratitude is extended to each person who has taken a moment to participate in this significant event. Thank you all for joining us today. Let's make this webinar an insightful and enriching experience for everyone involved. We have our keynote speaker, Dr. B. Prakash Rao, with us. Welcome, sir. Your insights would be highly valuable, and I would appreciate if you could present them in a speech. Over to you, sir. Sir, you can unmute yourself and share the presentation.
Yes, sir. You are audible?
Right, sir. Audible, sir.
Audible. So, shall we start or what?
Yes, sir. Yes, sir. You may start, sir.
Yeah, I'll.
You can share the presentation.
Yes, sir.
Right, sir. So make it a full screen, sir.
Yes, sir. I made. Yeah, is full screen, sir?
Yes, sir. It's for full screen, sir. Perfect, sir. Sir, please unmute yourself. Your mic is mute. Sir, please unmute yourself. You are not audible. Sir, please unmute yourself and make full screen. Your voice is not audible. Sir, voice is not audible. Please unmute.
Yeah,
Yeah, right.
I
Now,
unmuted.
yes, yes, now audible, sir. Sir,
Now
please make
audible.
right, right. Please make full screen, make it full screen, and then you may start.
Yeah,
Yes, right, sir, right. You may start,
right,
sir.
sir. Am I
Right,
audible?
perfect. Right, right, audible, sir. Perfect.
Audible. Oh, thank you so much, sir. Pradeep, sir.
Thank you, sir.
Yeah. Good day to all. Today, my topic is data-driven drug design, merging pharmacokinetics, DOE, and intellectual property strategy for smarter and research and development. Myself, Dr. B. Prakash Rao, and I am from Pharmaceutics. So, this grant, I got it. It is a mixture of chemistry and the Sutix. So that's why I was interested to show that I'll be telling about the same thing in coming slides. But patent was interesting. I mixed both the Sutix and chemistry. It was awarded. I'll be telling about this patent in the coming slides. Then the data driven, we had to take so many data from so many what you call genomic data, personal data, data collection from the different the real data, chemical data, all should be taken to have a advanced algorithm. That means you should have a process or principles which solve the problem to predict the modeling to accelerate identification of promising drug candidates. So here genomic, the human genome, it can be many patients, so it differs. It may differ from country to country, state to state, and so many. So for that reason, you should have the genomic data. That is very important because patients are different. Now, the everything just it was advanced. Individual therapy has come. Now, one drug you cannot give same way the same drug to the all people. The individual, the therapy has come into picture. So that is very welcoming step. So here the clinical data also very important. So you can take hospitals, health records, patient histories, clinical data. When we are submitting the the drug to the FDA or any authority. So that's what the patient demographics, whether it's from which one, all those things are required to give a one design. The next one, individual therapy. I told you already, the individual therapy has come. Now we cannot take even the T cells, they do by taking the individual technology and genes, then only they will do that treatment. The chemical data is also important, like the structures, properties, identification of potential candidates, their interactions with the biological targets, also very important to design the drug. So I told you already, the biological data, proteins, even most of the enzymes are proteins. So here, the proteins and genes, all those are identified. So really target. So many enzyme inhibitor drugs have come. So the mechanism, we had to know the target. That means enzyme may be a protein, may be genome, gene, or whatever it may be. So for that reason, we should have all the data before inventing the drug. So like omics, genomics, proteomics. The proteomics means which protein will be synthesized by genes, or what is the gene, how these genes are responsible to produce one specific protein. So for that reason, we should know the genomics. I told you already that the real world data, like patient reported outcomes, the real world evidence contribute to deeper understanding about the disease dynamics, how the treatment is going on, what is the progress, what is the outcome, all are required. So this data integration platforms tools are used to clean, harmonize, analyze heterogeneous data sets, enabling research to drive meaningful way. So generally, anyone, we had to discover the drug, then we had to develop the drug. There are so many types of designs are available. The ligand-based and structure-based design, the basic concepts of design, quantitative QSAR, that is quantitative structure activity relationships. Anyhow, I just briefly I'll be giving the most I'll concentrate on the pharmacokinetics and other. The structure ways here, the quantum mechanical approach. So quantum mechanical approach will tell you what is the interaction between the molecules, maybe covalent bond, or maybe a hydrophilic bond, maybe the bond with the protein, or docking systems, all are available. In a similar way, molecular orbital approach also, interaction between the molecules will be telling. The molecular connectivity approach, using the molecular structure, their property to guide the design of new drugs. So here, even another approach is also available, that is linear free energy approach, using the molecular structure to predict properties and guide the design of the node. So the design analogs, so many derivatives are available. Once upon a penicillin was invented by Fleming. He has been, after so many penicillin, benzyl penicillin, amoxicillin, amoxicillin, like that, so many. So when you know that the analogs, we can do the work. And there are lead compounds. The lead compound, cyclosporins, the lead compound, and the penicillins. In the similar way, so many targeted drugs, monoclonal antibodies, those are the lead compound, and small modification from that, we can make the better way of designing the drug. So here I'll give some example. Ritonavir and saculavir, those are created by structure-based drug delivery design, is to develop the protease inhibitor for the HIV. In a similar way, the anti-cancer drugs, imatinib, that is a key example, the design to target specific protein, BCR and ABL involved in the cancer. So this is about structure-based. The ligand also, monoclonal antibodies are ligand, hormones are ligand, genotype, gene fractions are the ligands. And so many ideas. So even levodopa, I'm going to tell you about the levodopa. I don't know whether people are explored or not, but today I'll give you the levodopa can be used as a ligand. So here, the cimetidine antihistamine receptor antagonist was started, and the cimetidine, ranitidine, all are produced, and antidepressant. So the serotonin responsible selective serotonin reuptake inhibitors are another example of drug design using the rational drug design. The key aspects of the drug design, the target identification, I told you already, protein or enzyme or the receptor, all those things we should know. When you know the target, then only we can design the drug. Even some of the, I told you, lead compound identification, cyclosporins and so many sulfonamides. Those are the lead compound. After that, modifications here, even penicillin is a lead compound. So how we modify each other to the better way, that is one. So that's what modifying the lead compound is the improve the binding affinity and desirable solubility, stability, reduce toxicity. So even the structure-based drug design, we are using the three-dimensional structure to target the design molecules. And even I told you that so many ligands are available. For example, the breast targeting or any targeting monoclonal antibodies are available for the cancer. Even my patent, I have taken hormone as a ligand, and the fractions of genes also taken be as a ligand. The ligand will transport as a carrier, will act as a drug targeting agent. So there are enzymes, maybe anything which will take to the target, those are called ligand. Of course, all the principles, instead of doing manually, we can give feed into the computer-aided drug design. But in this also taking the same computational tools to model, but we can we can rapidly design, and we can find out, we can reduce the lab cost, time, and we can save money and time and work. So that's what the computer-aided drug design also very useful. Here, so here today I'm going to present very important one, that is what we call properties like absorption, distribution, metabolism, and excretion. That's called ADME, CADME, or here. So here, bioinformatics also, we are utilizing computational tools. That there's so many, so much even genes and all the information are important. So that we can predict the and accelerate drug delivery process. Here in the similar way, this I explained already, and structure as activity relationships. Here, the isoelectric groups are available, like O2 minus, F minus, that sodium plus, mean they all have 10 electrons. CO, N2, Cn minus, NO plus, all have 14 electrons. We can substitute. Whenever in a molecule oxygen is there, we can substitute with CO is there, we can substitute the N2, or we can substitute Cn. So we can change the structure and little bit. So that's what those are called isoelectric groups. So those are also can be taken into that one. And so the much might talk about this one, ensuring the design molecule possess property like suitable for use. It should be useful, because so many patents are coming, but all are not coming into the market because so much suitability, because we have to be very accurate, we have to be very efficient. So for that reason, we should know about the ADME, absorption, distribution, metabolism, and excretion of the drug. For example, gold nanoparticles are available. They're wonderful, but they cannot be eliminated. And at the same time, they cannot be metabolized also. So for that reason, we have to be very careful in this. So here I told you already. So high throughput screening and combinatorial chemistry. Here the library, so many chemical library available. So that we can take and we can screen, and structure that resolutions also we can take. And today I will concentrate on the pharmacokinetics. What do you mean by pharmacokinetics? What body does to the drug? That's called pharmacokinetics. So the one is the absorption, then distribution, then metabolism, and excretion. At pharmacokinetic tells us the quantity, how much drug has come to breast, how much drug has come to blood, how much drug has come to the tissue, how much drug has to come to wound. All the pharmacokinetic that will be very useful. So it talks about the quantity and the movement. Kinetic means the movement of the drug. It will how it will move in the body, which tissue to which move, whether it crosses cell membrane or whether it crosses blood-brain barrier. All we have to consider. And then only we can know. So that is important. So here, the briefly, you may be knowing, but briefly, as my responsibility, the absorption, the drug in the dosage form, that will leave us out and come into drug solution, then enter into the gastrointestinal membrane, that will come to the blood. Systemic circulation, it will come. Immediately after coming into the systemic circulation, the protein binding will act. Actually, the body wanted to protect the foreign material. Drug is a foreign material. We cannot entertain into the body. For that reason, so many proteins are available, they bind the drug. But fortunately, this binding is reversible. So once the free drug is eliminated, the bound drug will come back. So that's what the protein binding and free drug will come out, and a lot of storage in the blood will be available. And after that, because as it is a foreign drug, the first reaction of the body is to eliminate. Maybe metabolism, maybe excretion. The first reaction of the body is to eliminate the drug, because it's a foreign. If you are taking amino acids, or if you are taking the glucose or anything, that will be assimilated by the body. But unfortunately, we had to give drug, but the drug is foreign material. So for that reason, we should know the pharmacokinetics of the drug. First, let us take the absorption. First rule, the drug should be unionized and soluble. Only soluble drug will only be absorbed. If it is in a particle, that cannot be absorbed. So for that reason, that too, it should be unionized also. It cannot, ions are not that much of the very less ions are absorbed. So another interesting, when you cross from stomach to the colon, the hydrogen concentration, one crore times it reduces. 1 crore times it reduces. So we cannot imagine that much change. So for that, we have to consider when you are taking a designing of the drug. And from the data, it was clear, very clear, pKa value of acid drug should be 4.5. Ideally, the basic drug should be 8. The ideal, of course, this can be this side or that side, but it should be 4.5 for acidic drugs, and basic drug is 8. Wonderfully, those will be absorbed. The most of the time, the molecular weight should be less than $600. Maybe 1,000 also can be allowed, but around 600 is wonderful. But here, after knowing my patent, I have seen the monoclonal antibodies, the molecular weight is 1.5 lakhs. Very good. It targets, it's specific, but does not cross into the tumor, does not cross the cell membrane. It is very difficult to cross the membrane. So for that reason, they add physiogenics to be given. Again, it already monoclonal is very costly. And again, we have to use another technology that causes another cost. So why can't you, the molecular weight of the monoclonal one is around 1.5 lakhs. Why can't they design a drug which is having only $600 tons? And similarly, and so much engineering was done about corticells. The corticells also used for drug targeting, but the only problem is the molecular weight are very big molecule. They cannot print it. It was shown the literature very clearly. The monoclonal antibodies and the corticells, they have good property targeting and other, but they have disadvantage of suffering. They're unable to cure solid tumors because the drug has to penetrate into the solid tumor. That is the most difficult. And as I told you, the pKa value, now I'll tell you the log p value. The log p value is the partition coefficient. So that should be ideally 3. It can be decided, that's why. But ideally, if it is 3, wonderfully that will be absorbed. So the K, the partition coefficient, so the concentration of drug in the octanol divided by concentration of drug in the water. So whatever, when you are designing, the structure should be balanced way. One is balanced, oil solubility should be there and water solubility should be there. We cannot compromise on it. So that's why oil solubility will be around 1,000 times than the water solubility. So for that reason, the log value, the p-value is 3. That means 1,000 times. So it should balance. So that's what we are telling. How do you balance? So lipophilic drugs means carbon chains, or the nitrogen or carbonyl ether groups are oil soluble, whether water soluble, polar groups like ions and carboxylic acids. So those are the groups we'll be telling. I'll be telling about those things. I have taken from the literature that is highly absorbed drugs. Highly metoprolol, propranolol, diltiazem. I have given the structure. You can see that one. Ether structure is there, amine structure is there. So much because the pKa value of the metoprolol is 9.67. So for that reason, it is well absorbed wonderfully. And even I observed the basic drugs, so that the basic drugs are more promising than even the naturopathy also says that it drink water from the copper because slightly alkaline. The slightly alkaline will be favorable for the good health. So here, why the metoprolol? Here I am showing the structure. There are ether group and amine groups are there, and that's why it absorbs very well. So we should see that ether groups, the neutral groups are very important. Another example, diltiazem, here also ether group is there, amine group is there, ketone group is there. The ether group is a neutral, pKa is 8.18. Ideally, very, very ideal pKa value as the basic drug, I told you about 8. So pKa value is 8.18. Wonderful drug. So that's why diltiazem absorbs very, very well. And now I'll tell you about, because I have done a patent on the prodrug. So prodrugs are, I'm going to tell something about in the prodrugs also. You can prepare ester or amide or amino acids are better. But amino acids are, because amine group is there, carboxylic acid, COH, carboxylic group is also there. So these will give the solubility very, very well. So for that reason, the prodrugs are, but phosphate salt solubility. Alcohol group is very, very important because metabolism is easier. As the metabolism is easier, toxicity will automatically be less. I'll be giving an example for that one. Amine groups are more important. So even my patent also, because I'm giving the, already I got granted my patent, it is based on the ligand-based drug design. In this case, I have taken estradiol. Here estradiol, the nucleus has the interaction with the receptor of the breast, the female receptors. So that's why, but you can block the OH or the CO group can be blocked. But the nucleus has the activity. So that's why the OH group are joined with the citric acid. And in the similar way, I have taken fludarabine as a drug. Here, fludarabine, only one amine group is there. I am joining with the citric acid. And it is a prodrug because fludarabine is already known drug. It is also orphan drug. It is also known drug. So we are joining with the estradiol. The estradiol is a ligand or targeting agent. It is a targeting agent. It targets to the breast. In the DHT, like dihydrotestosterone, target the prostate, the male hormone. Okay, so when you join together, if you link by other, very difficult because it will become NCE, new chemical entity. So one of my my guide, she suggested you make ester. So that's why I made citric acid, 3-carboxylic acid groups are available. One I made with the estradiol, another with the fludarabine, and so one COO group I just kept acetic. Citric acid is good. We are taking citric acid may not be toxic. So for that reason, I selected the citric acid. Here the citric acid has a 3-carboxylic group. The most of the problem with the anti-cancer drugs, they are bitter in taste. They cause nausea, vomiting. Again, we had to give another anti-vomiting drug. So I thought I'll keep the COH group as it is. When you are keeping the COH group as it is, it will give the sour taste. It will give sour taste. So that's the reason instead of succinic acid and malonic acid, I want to show, I selected the citric acid, 1-carboxylic acid may be free so that sour taste will be given. So this is about my patent, and I applied the yes patent, the application number already 1.5 years over. I'm waiting for the patent to come. And even I have so much chance of patenting my molecule. Even I can patent the bulk technology. I can apply for the dosage forms. So that's Indian patent number 502261. So this is a prodrug. But when I told you already, of course, I'll be telling about what are the IP strategy must be used. If you want, you can raise the hand so that I can understand. You can disturb in between. No need to one-sided. You can disturb. I won't be disturbed. I won't be disturbed. I can answer on the spot. No need to wait in the last moment also. You can raise your question or anything. And the absorption is over. The coming to the distribution, the measure pharmacokinetic parameter, that is nothing but volume of distribution. If the volume of distribution is 5 liters, that means that the drug distributes only in the blood. When the volume of distribution 10 liters, it will distribute in the blood and intracellular fluids only. If it is a 40, it will distribute in the blood, intracellular fluids, extracellular fluids. And if it 1,000 to 10,000, surprisingly, so much high volume of distribution. Of course, that much volume of the body will not be there, but that much volume, because this is called imaginary volume or hypothetical volume. So when it 10,000 of life, so it will go to blood, intracellular fluids, extracellular fluids, etc. Wonderful example is anti-malarial drugs. Anti-malarial drugs, they go each and every part of the body. So that's the reason anti-malarial drugs, fluoroquine, chloroquine, piperacquine, these are quinine, these are given 1 week a dose. 1 week a dose, because they distribute very largely. The volume of distribution is very, very high. So in and compliance, the patient compliance also increased wonderfully, because 1 week only, you have to take after 1 week only another dose. So I'm going to explain in the same that way. So here, the another interesting point here, the lip, if you have volume of distribution is high, then the off-life is very high. So most of the Sutix people, they won't do the what you call controlled regulatory system, prolonged regulatory system. If you create the drug itself a prolonged system and controlled system, no need another one. So that's what I'm doing. There are two distribution of life, elimination of life. Those are the pharmacokinetic parameters that tells you about the 50% of drug distributed and 50% of drug eliminated. These are the pharmacokinetic parameters. The wonderful observer, I was telling very clearly, the basic drugs are very, very interesting, because those are very useful, because they have high volume of distribution. That's why they are longer half-life. The longer half-life. So better, instead of ester, amide is better. In acidic drugs also, affinity to the albumin and lower lipophilicity than the neutral, and that's why. Acidic drugs are likely to bind only albumin and remain only in the plasma. So that's why they have lower volume of distribution. So for that reason, off-lives of the acidic drugs are very low, very low. They're very low. So for that reason, I told you already, basic drugs are wonderful, neutral drugs are wonderful. Those better design in that fashion. And lipophilicity of drug has a higher lipophility, and volume of distribution also very high. It will go to adipose tissue, proteins, tissues, all it will bind. I told you one wonderful example is anti-malarial drug. So here, the lipid, if the when the lipophilic drug is there, higher volume of distribution, higher half-lives. So that's what will be. If you have hydrophilic molecules like prost, the lipid membrane, even the membrane, the whatever membrane, cell membrane is a lipid bilayer. It will have the property of lipid in nature. The lipid in nature. So even if you take the oil, somewhat oily, that will give the taste, because the drugs which are soluble in oil will be very easily be absorbed by the system. The crosses the cell membrane. So better have the lipophilic drugs than hydrophilic drugs. But it should have balance, because completely we cannot take only the lipophilic. I told you already, the balance should be lipophilic should be 1,000 times, the hydrophilic should be 1. Okay, so that's what a log P is equal to 3. If it is a log 3 is 3, then will be partition coefficient is equal to 3 log P values. Then naturally, the drug is the wonderful drug it will become. And this was I was explaining. I am giving the example. The chloroquine volume of distribution 200 to 800 liters per kg. If you take ideal body weight 60 kg, 60 times, so more than 10,000. Because the basicity of the chloroquine is responsible for high volume of distribution. So that's why off-life is 20 to 60 hours. And due to the long chain of the lipophilic drug, you can see that very big long chain, and that's why it's causing the more distribution. I told you already, in the similar case, McQuilling, then pinning, or then piperaquine, all those things, example of anti-malarial drugs, they go on, they distribute. I told you already, 1 week once only we had to keep. So that is the advantage of the protein drug and high volume of distribution. Even some of the psychoactively acting drugs like risperidone, the respiratory half-life is 20 hours and volume of distribution is 60 liters. The basicity is a response. You can see there is no, when you see the structure, there is no acid at all basic drugs. So that's why the basicity is very, very important. And I told you already, wonderful. Once upon a time, the phenacetin was the analgesic and antipyretic drug. But here it has the ethoxy group. Ethoxy group. So it will be metabolized into paracetamol in the body. Then the researchers, they have taken metabolite of phenacetin, paracetamol. The paracetamol only drug, as it is made it a drug. So naturally, the paracetamol, you know well how much wonderful drug because of the free OH group. But introducing the hydroxyl group on the phenol is not so easy. But even then, the people have made the technology. That's why hydroxyl group is more important because very easily methylated. It can be methylated. It will be metabolized by methylation. So methylation is easier. As it is easier, it reduces the tox. It will be metabolized. As it is metabolized, it reduces the side effects. That's what. So phenacetin has gone and coming to the paracetamol has become wonderful drug. But no drug is beating like antipyretic like paracetamol today. And distribution, even I told you already, thiopental, wonderful example I have taken from the literature. It crosses the blood-brain barrier, highly lipophilic, onset of action is very fast. Within seconds, the person goes into sleep. So that is the thiopental. So I told you log P around 3, but it has a ideal log P value 2.9 and 3.56, very, very near to the ideal value. So that's why thiopental is used for so many surgeries also. Okay. And here I'm proposing one, I don't know in the literature, the levodopa. It is also polar. Polar substance cannot cross the blood-brain barrier. But there are the levodopa will be absorbed into the brain by active absorption. There are some carriers are available for taking the L-dopa. One of my classmates, he told nilotinib has a problem of reaching the blood. I told him we can join nilotinib with L-dopa because L-dopa will be joined by the chemical, some prodrug. I am proposing L-dopa is a ligand. Even I wanted to patent. Anybody who is interested, you can patent the L-dopa link with the nilotinib. The L-dopa takes the brain. Automatically, nilotinib also will be taken to the blood, into the brain, into the brain. So it is a wonderful idea. Alzheimer's disease, L-dopa can be used. Wonderful. It is a ligand. Maybe it is the amino acid, but to brain it is a ligand I am proposing. And there are some new recently donanemab, solanemab. So these are the clinical trials they are using for Alzheimer's. Now even the Alzheimer's disease, now challenging is Alzheimer's disease because many people are suffering from this. So it is the viable, financially viable area, Alzheimer's disease. So I am proposing L-dopa can be linked with the nilotinib so that the L-dopa will act as a ligand. Ligand. Wonderful. This is what you can take. This is the structure of the nilotinib. Here I am showing this is the structure of nilotinib. If you join with L-dopa, wonderful work can be done. And here, the morphine derivatives, how the group, of course, the basic drug like barbiturates or penicillin structure, penicillinic acid, all those basic drug that should be decided by or designed by the chemist person. As a pharmaceutics person, so small groups will make the other things so differently. Here I'm giving one example. Heroin, so acetyl group is there, whereas morphine, hydroxide groups only there, and codeine, only methyl groups are. I'm showing the heroin structure. Acetyl group, half-life in minutes because of estate group. The estate group is one that is causing in minutes only. When you take the morphine, there is no drug half-life is 2 to 4 hours, whereas codeine, we are using one methyl group. Because of that one, it is an antitussive. Codeine is an antitussive, and half-life is 3 to 4 hours. By changing methyl, acetyl, and free, so we can change the duration of action, and so that's what I'm proposing. So small groups, when you are changing, it will go on change the designing of that drug. And even off-life and metabolism, we are continuing with the metabolism. So here ester type of local instructs are available, amide type of local instructs are available, but ester types are having lower the off-lives, whereas the amides have the longer half-life. I'm giving example here because the esters are easily metabolized, whereas amides are slowly metabolized. So, and immediately, naturally, the half-life increases. Okay, so here I'm giving one example. The benzocaine half-life in minutes, it is also ester. Procaine is also ester. Half-life in 2 hours. Lidocaine, it is a amide. It is about 2. Bupivacaine, actually another example, bupivacaine, that is a amide. Because of amide nature, 8 hours the half-life. So, and less toxic also. So that's what we are saying that one. And I'll give another example, barbituric acid. How barbituric acid, acid structure we cannot change, but we can change R1, R2. So when R1, like allobarbital, we are using allyl group and methyl group. So allyl structure, because so it is a short acting. When you convert to methyl and isopentenyl barbituric, it is intermediate acting. When you take the phenyl and the ethyl, the only we are changing the R1, R2, one small group, so it is a long acting. So phenobarbital, you know very well, wonderful drug, it has been used in the common use. So because of phenyl and ethyl, it is long acting. It is for anti-epilepsy, is a wonderful drug, phenobarbital. So small change. Barbic acid structure we can't change, but we can change the even I told you that one. And I'm taking even the halogens also. Halogen, whenever you are designing, introduce halogens. So that's why lipophilicity increases, distribution increases, and half-life increases. And most of the halogen derivatives are like chloroquine is more active than quinine because of the chlorine. So efficiency increases. Toxicity may be more, but the halogens also can be used for that one. Here, the metabolism is somewhat difficult, and those halogens are can affect elimination from the body. Here, the halogen bonding will formed. So halogen can form halogen bonds with protein, which can enhance binding affinity. So that's why therapeutic efficacy is very high with the halogen. Like halogens means fluorine, chlorine. 5-fluorouracil, fludarabine, that's Fluorine derivatives. They are fluorine derivatives. They are wonderful drugs because they are anti-cancer drugs, anti-metabolite, anti-cancer drugs. So that's why halogens also can be considered when you are designing the drug. And of course, it will be oxidized, reduced, and okay. And here toxicity may be more, but some problem, environmental problem will also will be there with halogens. So we have to take care. And in summary, halogens are valuable tools in the drug, but they're important pharmacokinetic, the drug property. They affect drug absorption, distribution, metabolism, execution. And wonderful, I can say, efficacy increases, safety may be some problem, but efficiency of halogenated compounds are much more. To try to add the halogen. And the last one that is called in the kinetics, the elimination. Whatever drug is absorbed very rapidly, eliminates also rapidly. So that's why thiopental or thiopental sodium, it will absorb very fastly and eliminates. So that's why off-life is only minutes, 90 minutes or 5 minutes. So that's why the elimination execution also very, very fast. So that we have to make whatever the principles are governing for absorption of the drug, the same principles will be governing for excretion of that drug through urine or whatever it may be. Even I told you already the carboxylic acid group. I told you in my patent, I'm keeping one carboxylic group, one carbon free, because free carboxylic acids are sour in taste, we can reduce the bitter taste. So here carboxylic acid group adds lipophilicity and polarity. This may impede the bioavailability. But most of the anti-cancer drugs are suffering from what do you call vomiting sensation and other. For that reason, in my patent, I want to use citric acid. I want to take one carboxylic acid free. So that's what I have designed. Of course, it has to come into the market that I have designed. And that's what I'm proposing. If it is an amide, if it is a nitrogen, if it is a neutral, then the drugs are naturally sustained and controlled. No need to, we can, acidics are less possible, whenever possible. Not psychotropic, psychoactive acting drugs also, very high off-lives. So when you design properly, we can design for long. And I observed here, most of the antihypertensive drugs are have first metabolism. Because here I'll show you the cytochrome engine. It does hydroxylation, epoxidation, oxidation, demethylation. So try to see when you are designing the graph. So try to see less oxidative groups, and so that you can avoid phosphorus metabolism. That's what I was proposing. You design. Even I told you, when it is or is benzoyl penicillin off-life is 3 months, not stable. And when you convert amino acid, amoxicillin, off-life is 6 hours, unstable. So by changing the substitution derivatives, substitution groups, we can make the kinetics will be easier. And as in my topic demands, how to use a design expert. So here, whenever you want to, I told you hydroxyl groups, amine groups are very important. You select, so like here I am given hydroxyl group, carboxylic acid group, amine group, halogen group. So you consider as a categorical factor. There is no numerical. These are categorical factors. Those are called independent variables or factors. So, and in the similar way, now we can, whenever you want to design, so I am giving here, if you design log p values, The molecular weight, pK values are very important according to pharmacokinetics. Then optimize. Here I'm giving one example here. And here, so design expert, there are so many designs are available, like Placket Burman design, Taguchi. Taguchi designs are so well because here how to design, I'll just explain. I wanted to show design expert, but unfortunately that was in Hyderabad. I was unable to share. Here, how to select categorical factor we can select, numerical factor we can select. The categorical factor OHE in the molecule. So the low level, no hydroxyl group, high level with hydroxyl group. Or you can have a numerical, the number of, you have only one hydroxyl group or you have three hydroxyl. So you design in that and put into design expert and find out the log P values, pKa values, and how to see that one. And in the similar way, the amide group and methoxy group, all we can do that. And the machine learning software, 4.5 crores. Of course, if you are using the design expert, I told you by using this design, Taguchi, especially Taguchi is interesting, Taguchi design. So we can select categorical functions. Categorical means the hydroxyl group or what do you call amide group or ethyl group or methoxy group. Those are called categorical factor. Or when you want numerical, but in the design, it is wonderful. We can use categorical factors and numerical factors also. Or number of the two hydroxyl group, one hydroxyl. So like that, we have to use a design expert, and we can have a conclusion before going in. The basic research can be done before only into the market. So design expert software costs about too many, and 1 month free trial also available. Of course, it is a small technique, I'm telling. There is a small technique. By using the design expert, we can reduce the cost. We can select whether you want one or two, whether you want a hydroxy group or not. We can decide. And so here, Taguchi is advantageous because more factors. The factors means the functional groups, OH group, and less number of. For example, if you are taking the factorial design, there are three factors. Then 2 to 4, 5 factors. 2 to the power 5, 32 experiment we had to do. But when you are using Taguchi design, 1 or 3 factors, we can do with 5 experiment only. Instead of doing 32 experiment, we can do 5, 6 experiment. So that's why I suggest you can you can contact the pharmaceutical person who are well versed with the design expert. Even I'm good in the design expert. You can contact me for the how to use the design expert. Like Taguchi designs can be used to reduce the cost and the time. And whenever, but the intellectual property, but I'm not into the chemistry. Novelty should be there. Very easy to get novelty, but non-obvious inventive stuff should be there. And it should have the application. It should have application. Because without application, nobody will get the patent. But derivatives, no patent. Derivatives, no patent. But you should have your unique molecule, then you will get the, and structural uniformity is also required. We suffered structural uniformity when we applied. You can create product. Even my drug, I told you already, I combined estradiol and fludarabine by the ester group. It is a product, but it is efficient than the basic drug. So that's why I got patent. If you show patent, if you are product, you will not get patent. But if it is potent than high efficacy, then you will get the patent. So for that reason, I'm telling intellectual property, you create patent product which is more active than the drug. Then you will get patent. Automatically you will get the patent. So that's what I want to suggest that one. And very interesting point I observed. If you are taking new because why I have done ester group? Because ester group is a product. If you link by CHD group or any group, that will become new chemical entity, NCE. The NCE, if you wanted to get into the market, 1,000 crores we had to spend. 10 years we had to spend, lot of work, exhaustive document. If you are creating the product, wherever possible, create a product, it costs about 25 crores to 30 crores, and 5 years only, less work and less documentation, because the product easier to get, and same benefit you will get not all the products which are having efficacy get a patent. So I got for breast cancer and prostate cancer, I got the patent because of the product which is having more efficiency than the drug. So likewise, we plan for product so that we can do patent and marketing and money. And so if you have any questions, you can ask me, because I have given the I thought anyone will ask in meeting. No problem, you can ask. If you have any questions, please ask me. I'll answer.
Thank you so much, Dr. B. Prakash Rao, sir,
Thank you,
for
sir.
your insightful and enriching session on data-driven drug design, merging pharmacokinetics, DOE and IP strategy for smarter R&D. Your expert presentation beautifully bridged scientific depth with practical applications, providing the audience with a comprehensive understanding of how data analytics and strategic planning are transforming pharmaceutical research. We truly appreciate the way you have simplified complex concepts and shared real world perspectives. It has been an absolute privilege to have you with us today. Thank you once again for your valuable time and contribution to the SPSR platform.
Thank
Thank
you,
you,
sir.
sir.
Thank you, sir.
So, there are some questions from the participants.
Sure,
The
sir.
first one is how do the ligand based and structure based drug design approaches differ, and when is one preferred over the other?
Yeah, when the targeting known, structure based is given. Target is
Okay,
known, then only we can do for that one. Structure based.
okay. Thank you so much, sir. The second one is what is the significance of using bioinformatics and computational tools in computer-aided drug design?
The computational, because the library chemicals, more information is available. So even because whatever the bioinformatics, nothing but the target may be enzyme or protein. Naturally, whether we can see in silico only, we can see whether your drug is acting on the protein or on the docking system is available. You can see that, and you can reduce the time. You can reduce the time.
Right, sir. Thank you, sir. The third one is how can machine learning and AI tools enhance predictive modeling in pharmacokinetics and lead identification?
Yeah, I don't have much idea about machine learning, but what I understood, if you give the structure, it will give you all the pharmacokinetic parameters within no time. But maybe
Right.
costly. Maybe costly because I told you machine learning 4.5 crores to earn 4 crores, it will take time, but you can reduce. But I heard when you give the structure, automatically it will give. Maybe those are acting on the what do you call multiple regression. Because if watch groups is there,
Right.
what is the pharmacokinetic parameter? If the CH3 group is there, what are the pharmacokinetic parameters? All will be predicted by machine learning. I have seen, but I have
Right,
not
sir.
used machine learning. But what they told, if you give structure, it will give all the pharmacokinetic parameters without doing any experiment.
Right, sir. Thank you so much, sir. The last one is, sir, can you describe the typical timeline and cost comparison between developing a product versus the new chemical entity?
Yes. Thank you, sir. Actually, if you want a new chemical entity, as it is a new chemical, we require preclinical, phase 1, phase 2, phase 3. Okay.
Right.
But if it is a prodrug, preclinical and the phase 1, no need to do. The preclinical and phase 1, it takes about 1 to 6 hours. Yes. So we are
Yes.
reducing. But as it's a prodrug, we can take data from FDA. The
Okay.
product data is available. We can submit. So preclinical and clinic phase 1 need not do. So we can reduce almost 1 to 6 years. 1 to 6 years we can reduce. So when you are reducing the cost also will be reduced.
Right,
And
sir.
time also reduces.
Okay, sir. Okay. Thank you so much, Dr. B. Prakash Rao, sir, for answering all the questions so insightfully and patiently. Your responses have not only clarified the complexities of AI in drug discovery, but also provided valuable perspectives for both budding researchers and professionals in the field. We truly appreciate the time and effort you have dedicated to this session. Your
Thank you.
contribution has made this webinar both enlightening and inspiring for all of us. Thank you so much, sir.
Thank you, sir.
So here we come. So here we come to the conclusion of this session, sir. As we come to the close of our insightful 98th SPSR webinar, we extend our heartfelt gratitude to our esteemed speaker, Dr. B. Prakash Rao, sir, Director R&D and IQAC, Moonray Institute of Pharmaceutical Sciences, Shadnagar, Telangana, India, for delivering an intellectually enriching session. Today's discussion on integrating pharmacokinetics, design of experiments, and intellectual property strategy in data-driven drug design has provided us with a deeper understanding of how multidisciplinary approaches are shaping the future of pharmaceutical R&D. The clarity with which Dr. Rao presented complex concepts from product design to predictive pharmacokinetics and IP frameworks was truly commendable. We are confident that the knowledge shared today will inspire researchers, academicians, and industry professionals to adopt smarter and more efficient strategies in drug development. On behalf of the Society of Pharmaceutical Sciences and Research, we thank all participants for their enthusiastic involvement and continued support. We are deeply thankful to Dr. Shashi Alok, the National President of SPSR, for honoring us with his presence. We recognize and appreciate your commitment and guidance, and we offer our profound thanks for your insightful input. We would also like to express our appreciation to our convener, Mrs. Monika Sabharwal, for her pivotal role in organizing this event. We extend our gratitude to all the SPSR team members for their collective collaborative efforts, which have broadened our understanding and fostered cooperative learning. Your collective contributions have made this webinar a success, and we look forward to continued collaboration in advancing pharmaceutical sciences and research. We look forward to welcoming to our upcoming webinars. Stay connected with SPSR as we continue our journey of learning and innovation in pharmaceutical sciences. Thank you and have a wonderful day ahead. Thank you. Thank you
Thank
so
you.
much, sir. Thanks from Team
Thank you, sir.
SPSR.
Thank you. Thank you, sir.
Sir, we may leave the session.
Okay. Fine. Thank you so
Right,
much.
sir. Thank you so much, sir.
Thank you so much.